Let's dive directly into Figure 4 of Osterfield2013 and use it to thoroughly analyze the study's implications, strengths, and future considerations. Figure 4 reveals the molecular signatures of mechanical tension during dorsal appendage morphogenesis in the Drosophila egg chamber. In these panels, the authors use immunostaining for SQH-GFP, which is a proxy for non-muscle myosin II, and bazooka, a polarity protein. The figure is central to linking apical tension with the observed cell rearrangements during tube formation. Now, we'll first shift our focus to Figure 4, Panel A, which is highlighted here in red. Focusing on the left panels from the main figure, Panel A, we observe that SQH-GFP, which marks active myosin, is highly enriched along the interfaces between the floor cells and both the roof and midline cells. This enrichment indicates that these boundaries are under considerable tension. The specific localization along these smooth, well-defined interfaces supports the idea that increased contractility here drives the bending of the epithelial sheet. This promotes the onset of cell rearrangements, which are essential for tube formation. Complementary to the SQH data, the bazooka signal tells a similar story, but from the opposite perspective. Here, bazooka is strongly downregulated precisely at the floor-to-roof and floor-to-midline boundaries. This complementary pattern, where regions of high myosin accumulation are proportional to the reduced bazooka levels, reinforces the notion that these boundaries are zones of high tension. Essentially, while SQH marks contractile forces, the dampening of bazooka underscores a local loss in the polarity or adhesive cues that might otherwise stabilize a flat epithelial architecture. As morphogenesis proceeds, the spatial distribution of SQH-GFP is not static. The later panels here illustrate that as the tissue begins to bend and cells initiate intercalations, the intensity of SQH along the floor to midline boundary reaches a peak near the site of active intercalation. This dynamic shift implies that the tension is being modulated in time to precisely orchestrate tissue remodeling. The clear correspondence between tension marker dynamics and morphological transitions strengthens the proposed model in which pattern apical tension drives the formation of three-dimensional structure from a flat epithelium. Ultimately, these observations have important implications. Figure 4 provides compelling evidence that pattern apical tension, demonstrated through localized myosin enrichment and complementary downregulation of bazooka, is a major driving force behind the morphogenetic processes shaping the dorsal appendages. A key strength of this study is its integration of high-resolution imaging with computational modeling to tie protein localization to mechanical behavior. However, one potential limitation to note is that the study relies on indirect indicators of tension. Therefore, future work might incorporate direct biophysical techniques such as laser ablation to further confirm these inferred forces. In conclusion, Figure 4 demonstrates that a two-dimensional pattern of apical forces can initiate three-dimensional deformation and drive complex tissue rearrangements. This work not only advances our understanding of the mechanistic underpinnings of tube formation in Drosophila, but also lays the foundations for exploring similar principles in other developmental systems. Future experiments that directly measure tension using approaches like laser ablation or optogenetics will be integral to validating and extending these findings. The integration of experimental data with computational simulations exemplifies a robust, multidisciplinary approach, one that can be harnessed to address similar morphogenetic questions in other contexts. Specifically, optogenetic tools have revolutionized our approach to studying dynamic signal transduction. By employing systems such as OptoSOS, we can oppose defined, reversible RAS activation patterns and directly observe how cells decode these signals. For example, here, Bugaj and colleagues used this approach, as well as a high-throughput platform called the Optoplate, to show that cancer-associated changes in BRAF not only elevate basal signaling, but also slow the decay of ERK activity. This altered kinetics causes the cell to misinterpret pulsatile inputs as sustained signals, ultimately driving inappropriate proliferation. Such insights highlight the tremendous potential of optogenetics for both uncovering fundamental cellular mechanisms and guiding the development of novel therapeutic strategies.